Welcome, this is the first video in a 3-part series on molecular docking. In this first video I'll be introducing you to molecular docking, what the basics are, and then I'll dive into the most recommended software for the vast amount of proteins that exist. And then I'll present my add-on to molecular docking using that software, which enhances the docking experience by tenfold. So with that being said, let's begin. So molecular docking. It is essentially just predicting how small molecules interact with proteins. We call these small molecules ligands, and we call the protein the target or the receptor often. So what you will encounter in the basics of molecular docking, it essentially consists of your target, as I said, the protein, which could be an enzyme, and the ligand, which could be our substrate. And let's say we run a docking simulation using an enzyme and its native substrate. Well, what's going to happen is that you're going to be finding the active site of the enzyme. However, it's not always going to be the case that the docking simulation is going to align with reality. Sometimes it will differ, and sometimes substantially. But molecular docking, despite these flaws, is still a great achievement for all of computational biology, and it's already accelerating how we are generating new data and expanding our knowledge of life. So the point that I'm trying to make is that molecular docking from a mechanical view is actually really simple. It's like Lego. You have one block and then you have another block, and it fits on top of this other block or beneath it or besides it or inside it sometimes. And that's where you can see we have this incredibly complicated three-dimensional structure of a protein down here. And I'm just going to use a laser pointer. So we have this protein, which is represented using its atoms and how it occupies some space in the real world. And you can see it has crevices, it has these valleys, and all of these areas are potential sites for some ligand, in this case this molecule right here. And by running the docking simulation, it correctly finds, or I should point out that it often finds the correct one, and that's why you have to run multiple of these simulations in order to fine-tune parameters and really figure out where is the most energetically favorable place on this protein or inside the protein. So that was just the basics of molecular docking. Let's get into something that's also very relevant, which is regarding the ligand position. So when we're talking about the ligand, there are two very important terms that you need to know about. So one of them is called pose and the other one is called conformation. And these two are very essential to the results that you're going to be obtaining from a docking simulation. So the pose is very simple and straightforward. All it is how is it oriented in space. I have made this little cysteine amino acid, and I'm going to use this just to visualize what I mean by this. So two different poses would be like, okay, here's one pose and here's another pose. So the key thing here is that the atoms themselves are not in a different position in respect to one another. That means this carbon is still slightly this way and up from this carbon. And that leads me to conformation. So a conformation referred to the different ways that these atoms can be arranged in respect to one another. That means, let's say that this sulfur group is slightly bent towards the acid group over here. That's one conformation. And another conformation could be, let's say it's oriented away and towards the amine group right here. So that is two different, maybe you can see it better here. So this is one conformation and this is another conformation. So during a molecular docking simulation, it is going to use different conformations that are energetically favorable and of course still obey the laws of chemistry and not breaking any covalent bonds or merging other. And by doing this, it can generate more realistic predictions of where this ligand would bind. So think of it as the ligand is somewhat being molded into the shape of the active site, which enhances the activity of the ligand. So what's very important to remember here is that the poses is just the number of different results that you get. So let's say you want to run the simulation 10 times. You want to get 10 different predictions of where this ligand would bind, and that would result in 10 poses. The thing is to remember that some of these poses might use the same conformation. That means one conformation could be let's say this cysteine is up here. This could be used by 4 poses, and then another 4 poses could be using this conformation, and then the last 2 poses could be using just like standard, like without me bending it. So that would only have 3 different conformations, but in total it would have 10 poses. I'm gonna illustrate here. You can see in this little video how the different poses and conformations are being swapped in the site that this docking revealed the most energetically favorable. So behind all of this elegance lies a lot of very interesting math, chemistry and physics. So I wanted to present what's actually being calculated under the hood when you're running such a molecular docking simulation. And as you can see, there are a few illustrations, a lot of equations. It's a very interesting field. Alright, with the basics out of the way, let's move into the more practical side. You want to find the software that you need to use. Okay, so when I entered into the world of molecular docking, I was faced with this one issue of metalloproteins. So a metalloprotein is essentially just a protein that consists of 1 or more ions somewhere in its structure, and where these ions are responsible for the function of the protein. So without these ions, the protein would not work. And we call these metalloproteins. And they are around 50% of all proteins we know of right now. So that means they're pretty much everywhere. And when I started my molecular docking journey, I was working with a protein that consisted of a calcium ion, and that forced me to find some software that accepted metalloproteins. And the problem is that most software does not accept it. If you do a quick search on molecular docking tutorial, the first one that you're going to find is probably AutoDock Vina, which is incredibly easy to use, but the problem is that it's not capable of correctly simulating these ions inside proteins. And generally speaking, they're pretty complicated to imitate in silicio. However, there is some software to use, and you may be asking me right now, well, what is it? Well, it's called AutoDock 4 by the Scripps Research Institute. And this is a great piece of software. It accepts these metalloproteins. It's a reputable software, been used in hundreds of articles. It has universal compatibility, which means it can run on a CPU core, and that makes it very easy to employ on pretty much any computer nowadays. Of course, GPUs are superior. However, for hobby / amateur and not real bioinformaticians like me and I think a lot of other people, this is a great solution. I need to point out that running AutoDock 4 on a Mac is not something that I've been successful with, and I think it's very difficult. I'm not even sure if you can do it. So as far as I know, you can only run it on Windows, and I have had great success on Windows, so I by all means recommend Windows. Also Windows 11, by the way. So that's great. Let's take a look at what AutoDock 4 is all about. So here's the flowchart of AutoDock 4 and how it's natively being used. So that means just like straight out of the box when you download it. So the first thing is entering parameters. It's like the grid box, the spacing, number of poses, aka how many iterations are you doing this simulation. And that's pretty straightforward. Then there's the grid map generation, which is done by a subsidiary program called AutoGrid. And AutoGrid will use your information right here, and it'll generate data that represents a 3-dimensional cube of atoms and like individual charges that are necessary for the docking to run. And then using AutoDock.exe, you can run the actual docking simulation, which is the main part of AutoDock 4, of course. And then number 4, you get DLG files. A DLG file is the output from a AutoDock simulation, and you can view these just by opening them using your notepad, because they're actually pretty readable for people. They're written almost like just a long Word document, and that makes them very easy to like analyze, and you can scroll through them, and then you can check what's the best interaction, the best conformation and pose. And then there's also AutoDock Tools, which is a part of the AutoDock environment, and that's the GUI, and you can import your DLG files into that, and then you can view each and all of the poses on the receptor. But that's just how you normally do it, so that's just for reference. However, here's the thing, there's a lot of flaws, so I'll just quickly mention them. So the first one is software freezes, crashes, and Python goes nuts. Essentially, these all encompass the experience that I've had, which was when using the GUI incorrectly, sometimes it would cause the underlying Python to essentially just break, and it was almost like it became corrupt. So you had to completely reinstall it, all of the software, and it was like 2 programs individually and a lot of files all over your computer. It was terrible. And not to mention the GUI, which is so hard to navigate. There are so many sub-menus, and it's a steep learning curve. And currently the only way you can learn about this is lengthy Indian YouTube videos online that all teach you how to go through all of these steps that I showed you on the previous slide. And it's funny because the Indian people are almost like exerting a form of monopoly. They're pretty much the only people on YouTube that are teaching you how to use bioinformatic tools. That's why I'm here. I'm trying to change that. And not just change the video format, but also the underlying problem with AutoDock 4. So let me present my solution to all of these problems that I've been addressing. So my solution is called LightningAutoDock Tools, for short LAD Tools. You can do docking in less than 5 minutes, and I'm not joking. It is incredibly easy using my suite, which is essentially just 6 tools that you can use to quickly crunch all of the preparation that's needed for a docking simulation. And then it consists of this complete pipeline from start to finish, from obtaining the ligands to investigating the results graphically. And it's all built around transparency, so it just consists of independent Python scripts for each and every step, so that makes it very easy to follow along and redo things if something goes wrong. And then one of the main selling points, well, this is not a selling speech by any means, I'm just trying to help the community, but the main thing here is that you can do large scale screening simulations. It does this by utilizing multiple CPU cores at the same time. So CPU parallelization, you can utilize all of your CPU's power and juice, and then run multiple simulations all at once, and that means you can grind through a list of ligands really quickly. And then I've worked really hard on the stability because it's running within the Windows environment, which means things can get kind of glitchy sometimes. So I've ensured that it's capable of running for plus 30 hour simulations. And then another really nice thing is the compatibility with Chimera X. So Chimera X is a molecular graphics software from the UCSF university, and it's a great piece of software for viewing interactions, and just it's very user friendly. So what I've done is that the results from the docking simulation that come in these DLG files, well, you can run this one script that I've made, which will then convert these DLG files into a AutoDock VNAV file. And the thing about the AutoDock VNAV file is not to get too technical, but they're special because Chimera X has this function built in so that it can import these AutoDock VNAV docking files, and then you can view the results very easily by just tapping your keyboard keys back and forth. And then I've resolved some of the errors that otherwise would appear using the command line interface. So that's just a general overview. So the LAD toolkit, it looks like this. It consists of 6 different parts, Chimera X being one of them, and then 5 different tools. So let's look at it. So on the prep side of things, we have L-Protein, LSAB. LSAB stands for Lightning Screen and Babel. And this LSAB program, it just combines L-Screen and L-Babel into one process, so you don't need to run them individually. So it just saves you time. After having run these 2, you'll have your ligands ready as well as your protein ready. So some of the protein preparation does take place inside Chimera X, but the ligand preparation is entirely done using LSAB. And then you can do the docking simulation, which takes place inside the LAD dock. All of these are Python files, by the way, so they have .py. And this LAD dock is the one that does the screening capability and just facilitates the entire docking. And you configure all of the parameters in a config file, in a txt file for this program to work. And then you have your results. So as I mentioned, I have this called LVNAN, which is compatible with Chimera X, so you have this wonderful way of viewing your results. Then there's also LView, which is a program that I made before LVNA that allows you to sort all of your results very quickly. So let's say you want to find the strongest match, the best candidate for, let's say, this receptor. And then you can just quickly find the top number 1 or number 5 or something. So that's a very easy way to sort in your results. Let's say you have hundreds of results. I'll need to mention that all of this runs within the Anaconda Python terminal. And I say this because Anaconda Python terminal is by far the most reliable way to run the LED toolkit, specifically because of this LBabel, because LBabel is utilizing the OpenBabel extension for Python, and installing that just into your normal Python you download from the official website from Python, it is full of errors. I could barely get it to work. So when using Anaconda, it ran smoothly. So maybe it's just my thing, but I highly recommend Anaconda. It's more optimized in general. And if you're wondering what this L stands for, it's for lightning. So lightning protein, lightning SAB, lightning screen, LightningBabel, LightningAutoDock. So it's just to abbreviate so you don't need to spell it all in the terminal when you're using these scripts. And Lightning, of course, because it's really fast. It's much faster than the native AutoDock for it. How did this all start? Well, it actually came out of an iGEM project that I was part of. I was on the team BKEA, which is Bikea or IKEA when we were in our funny mood. We were working on the project Renaultsign, which was about kidney stones. So iGEM, for those of you who don't know, is the world's largest synthetic biology conference / competition. I think there was around like 9,000 this year. It was incredible. I'm down here in this picture somewhere. Our team was wearing blue jumpsuits. But the takeaway here is that the software that I was just presenting you with, this LightningAutoDock Tools, was actually awarded the best software tool prize. I need to mention that I was in the high school category, which means that it was not in the university standard, but that's about it for this first video. I'm hoping I'll see you in the next part. That will be about the installation procedure and how you set up the environment and all the pitfalls, because to be frank, there's a lot that can go wrong. But I hope you enjoyed this video, and I hope you found it useful, and until then, peace out.